This is Campbell's Biology, 7th edition, Chapter 19, Eukaryotic Genomes and Regulation. And we'll start from the very beginning. Just some basic background on terminology here. We have something called chromatin, which is unwound DNA with its associated proteins, for the most part histones, that helps to pack the DNA into smaller compartments. And we'll get into the various types of chromatin that we describe. But imagine you have a chromosome, which is highly packed, highly condensed, only visualized during, only visible during mitosis or meiosis, and we call it just basic chromatin during all the other times when the cell is not in mitosis or meiosis. So basically, all during interphase. So we talked in 18 about how prokaryotes regulate gene expression in different environments, and here we're going to talk about eukaryotic DNA. So we already mentioned that it's associated with large amounts of protein. And they contain lots and lots of DNA compared to their length. So they're able to be packed into very small spaces. And so histones are the names of most of the proteins that the DNA gets wrapped around, almost like marbles that you wrap a string around. And that's what we call together DNA and histones is chromatin. And it binds tightly to the DNA. And when you take those histones and the DNA and you wrap them upon each other, the next phase of packing we call nucleosomes. So we have a bead. Each bead that you see here, where this is one bead, essentially, is a nucleosome, many, many histones. And then we get on even further, where this is one nucleosome and it folds back in upon itself, of course. And then we get these loop domains where the DNA is wrapped even further, where there's many, many beads along this one length of loop domain, and it's attached to this basic scaffolding protein. And then finally we get into the actual chromosome. Here's a chromosome in metaphase, where if you look down in this region of the chromosome, this is what we're seeing up close with the loop domains and so on, as we mentioned before. So in interphase, we don't have the chromosome. We just have chromatin, and we call it euchromatin, to be more specific. It's highly extended. And now we get into how the compacting of the DNA is regulated by various tags and so on. So all the DNA in every single cell is the same, carries the same genes, the same non-coding regions, and so on. It's just a matter of which genes are turned on that determines what the cell's function is going to be. And this is a very important component of cell differentiation as the organism develops from embryo into an adult. So each multicellular eukaryote expresses only a fraction of its genes. This unique subset of genes helps to differentiate or to distinguish what that cell is or is going to do. And there's regulation at various points, whether it be during transcription, post-transcriptional processing, as we talked about with the 5' cap, the G cap, or the poly A tail, and alternative RNA splicing, which we'll hit again, as well as regulation of once the mRNA is even produced, you can degrade it before it reaches the ribosome, and then the protein itself as well can be altered in its shape, can be paired with others, can be inhibited through competitive inhibition or allosteric inhibition, if you like. All right, so then we have a different form of chromatin called heterochromatin. Heterochromatin is usually not expressed. Heterochromatin is very tightly packed. And the way that we believe the conversion between euchromatin and heterochromatin affecting gene expression is due to histones. Histones have these tails that stick off of them, and there's something called histone acetylation. And histone acetylation is when acetyl groups are bound to these histone tails, and that causes the loose chromatin structure, and that promotes or allows transcription. When histones get acetylated, transcription may occur. When there's no acetyl groups attached, then it's very tightly packed. And we have heterochromatin, where this would be euchromatin. There's also methyl groups, which we saw in the epigenetics video from Nova. If you haven't seen that, I recommend going to Nova's website and looking at the shortened segment of the epigenetics piece. It's very interesting, and it talks a lot about methyl groups and tagging of DNA. When a methyl group is attached to DNA, it's associated with reduced transcription, or it turns genes off, methyl tags as they call them. And this we tie to epigenetics, which is basically the inheritance of traits that are not directly involved in a specific nucleotide sequence, or if you like, a gene. So it's the non-coding regions and how they affect the expression of the genes, both how the environment affects that expression and the individual itself. So then gene expression is regulated in a variety of ways. We've already done histone acetylation. We've talked about methyl groups. And we can alter the way that the transcription machinery binds as well, this being largely RNA polymerase and various control mechanisms associated with that we mostly call transcription factors, but we'll look at some of those here. So here we have a gene that we want to transcribe. There's your exons and your introns, of course. And just upstream of the promoter, which is just upstream of the gene, we have these proximal control elements that we'll talk about in just a second. Up here we have these enhancer regions, which are even further or separate from the proximal control elements upstream. And these segments of non-coding DNA help to regulate transcription that we'll look at here in just a second. They're largely tied with transcription factors. RNA polymerases require the help of transcription factors, which are proteins that either assist or block RNA polymerase from working and binding. So the proximal control elements are right next to the promoters, we said, and distal can be far away or even on a different chromosome or even in an intron. I should say not on a different chromosome, but within an intron, a non-coding region. So here we see an activator. Activators are proteins that are associated with the enhancer regions, the distal control elements. And when these activators bind, this triggers a folding by the association of a DNA bending protein. And by the way, this level of detail is not that important for the new College Board AP test, but understanding how these enhancers work might be a good thing. Enhancers associate themselves with activators. The enhancer are regions within the DNA upstream of the promoter. And this DNA bending protein bends these activators in place to associate with a variety of transcription factors. And those transcription factors enable RNA polymerase to bind to the promoter and to synthesize the RNA, to transcribe the RNA. These are activators associated with the enhancers. You can also inhibit the expression of genes, of course, with repressors. In much the same way, but preventing the DNA bending protein from binding or blocking the activators from binding to the enhancers and so on. Now, in prokaryotes, we saw operons, where one promoter is associated with a whole sequence of genes, but this is not the case in eukaryotic genomes. Eukaryotic genomes have one promoter for every gene, essentially, and with that promoter, there are also associated control elements, as we just saw. Now, if there's one group, say a sequence of enzymes all necessary for the same metabolic pathway, let's say in a human, the formation of tryptophan, similar to an operon that we saw in a prokaryotic cell, it's not laid out in an operon, but in a eukaryotic genome, all the enzymes of that pathway may use the same promoter, and thus the same transcription factors. And they're also often going to be found very near each other on the same chromosome, and we'll see that a bit later. So even after transcription, we can regulate the expression of a gene. Even though it gets transcribed, doesn't mean it's going to get expressed. Here we mentioned previously alternative or differential RNA splicing, where the essential thing here is what is treated as an intron can vary, and thus what is removed by spliceosomes can in one cell be considered an intron, and in another cell it might be considered an exon. And thus the actual coding DNA that gets sent to the ribosome to make a protein can vary or is different for the same set of DNA. You can also take the mRNA and degrade it, or to cut it up essentially, and thus affect or block protein synthesis. And this is often determined by the leader and trailer regions, the poly A tail, GCAP, and so on. Here's RNA interference, or commonly known in the field as RNAi. Now, RNAi is created by microRNAs which get cut up by a protein, and those microRNAs get associated with the protein that binds to the DNA and blocks the binding and the transcription by RNA polymerase and the various transcription factors, and then promotes its degradation, so that this gene will not get expressed. Even though the mRNA was made, the RNAi blocked it. Interference RNA. And that's essentially what's described there for you. And you can also simply, the initiation of translation itself, so following transcription, translation itself can be blocked by regulatory proteins that bind to the mRNA and prevent the ribosome from reading the mRNA. And then, of course, after translation, you can modify the protein itself by adding certain chemical groups and cleaving or cutting up the protein. As you see here, proteasomes are taking this ubiquinated protein, which are the tags, the trigger for, oh, look, cut me up, I'm of no use anymore. And then this proteasome chops it up and recycles the pieces into peptides to be reused to form another protein. And we'll take a pause on this one.